So DNA fingerprinting using PCR is a really important thing to understand. If you ever find yourself on a jury, or if you ever find yourself accused of a crime, or a loved one accused of a crime, it's probably a good idea to understand a little bit about PCR-based DNA fingerprinting. Like, this is also called DNA profiling. The word profiling bothers people, but it's really just using someone's DNA evidence to match them with something or to exonerate them from something, to identify someone or something. Right? PCR-based DNA fingerprinting can be used on humans. It can be used on every living thing because every living thing has DNA. So we can use this, for instance, if you did a gut content analysis, you could look at the bits and pieces that you found in the gut of a fish, for instance, and you could see based on the DNA that you extracted from that gut content what types of things that fish had eaten. So it can be done on all of the living world. What we're doing with this is we're not analyzing all of the DNA. We're analyzing DNA fragments and we're analyzing little pieces that we're targeting using PCR to determine, what the identity is of the individual. We use these things called genetic markers, and they're found in non-coding regions of the DNA. But there is variation between individuals, even humans, though we are very, very, very close, very similar genetically, there are still about 16 million differences. So we use PCR so that we can amplify this target DNA so we can better visualize it. You could theoretically visualize it if you had a small amount of it, theoretically, but it makes a lot of sense to amplify it, make more copies of that target DNA so you can visualize it better and have more convincing data. So again, this would normally have been done in lab. If you compare the DNA of any two people, they are about 99.5% genetically identical. There are small differences between any two human beings. It's still about, as I said, 16 million differences. So we can use those differences to differentiate, because I don't like that word, between any two people on the planet. Right? Those 16 million differences, even between siblings, would allow us to differentiate, okay, was it you or your brother who was the one who left DNA at the crime scene, for instance. Again, like I mentioned, we don't amplify all of the DNA. We look for particular things called short tandem repeats. Tandem is back to back, right? Short, you know what short means. Repeat, well, we know what repeat means. So we look for this repetitive DNA. It's non-coding DNA. Back when I was in college, it would have been called junk DNA, which again is coming in handy. And this repetitive DNA is present in multiple copies in the genome. Now, how many repeats, right? How many copies of that particular short tandem repeat are present in a particular individual can be characteristic of that individual. And so we have different numbers of those repeating units that can be characteristic to an individual, and we use this in DNA fingerprinting or DNA profiling. This is doing short tandem repeat or STR analysis, comparing the lengths of the STR sequences at these specific sites. Now, what does it mean by lengths? Well, let's say you have a repeat that is A, T, T, C, G. All right, now what that means, right, they're short tandem repeats. So let's say that is, we're just going to say it's this long. Right, I got to erase that now. It's annoying. So it's this long. Now, if you have only one, your DNA is going to be a very small sample. Right, if you have two, they're going to be tandem, so they'll be back to back. So when you cut that piece out, you're going to have a piece of DNA that is twice as long. Right, if you have three, and I'm going to try to do this here, 1, 2, 3. Right, they're tandem, so it's going to be 3 times as long as having one. So if you have 3 copies of the ATTCG STR, that sounded sexy, right? You're going to have a piece of DNA that is 3 times the size of someone who has 1 copy of the ATTCG STR. Because when you do this with PCR, and you amplify that part of the DNA, you will amplify a piece that is 3 times as big if you have 3 repeats. And as we learn in the gel electrophoresis part, we can separate a piece of DNA that is 1 repeat long versus 3 repeats long based on size. Now we don't do this STR analysis with just 1 STR site. At the time that this was published, which was a couple years ago, scientists were using about 13 different STRs, and we're going to look at some examples of that. So every living thing has these STRs, right? You can have 3 repeats or multiple repeats. This is the TCAT repeat, and you can see they're tandem, right, back to back to back. So here are some examples. Right at the top, we have this individual has 4 repeats, right? 1, 2, 3, 4. And this one has 6, this one has 7, right? These are in your chromosomes, right? So this DNA is inherited from you to your offspring. All right, remembering that you get 1 allele from your father and 1 allele from your mother, right? And you as a human, you're going to pass on 1 allele to your offspring. In this particular case, your copy of the chromosome, and your partner will pass on a copy of his or her chromosome. So if this is me, let's say if we're looking at my TCAT repeat profile, I have a 6 repeat and a 5 repeat. Right, here's my mother and my father. And what we can see is, okay, where did I get my 6 repeat? Well, not from Dad. Dad had a 4 and a 5. From Mom. All right, where did I get my 5 repeat? Well, not from Mom. She had a 6 and a 7. From Dad. So you inherit 1 from each parent. All right, we'll look at my grandparents. All right, where did my 6 come from? Oh, there it is. My 6 came from Grandma. Okay, where did my 5 come from? Okay, my 5 came from my grandfather on my father's side. There it is. So we can trace these back almost like a pedigree. Now, if we were to look at this, let's say we had DNA from a crime scene. We want to look and say, are there any 2 people in this family group that would have the same patterns? So if we were to take this DNA and run it out on a gel, are there any 2 people that have the same patterns? So let's look at mine, right? I have a 6 and a 5. So if we were to run this out on a gel, here's my gel. Right here are the loading wells. If we were to run it out on a gel, right, we'd have the 6 being closer to the well because it's a larger piece of DNA, and the 5 being right below it. Okay, now if we were to run my dad's on the gel, we'd have a 4 and a 5. Well, that's not the same. We're going to look at Grandma on my dad's side, 4 and a 7. No, that's not the same. Grandpa, he would have a 5 and a 6. All right, so Grandpa would have a 5 and a 6. So the lanes or the bands on the gel would be identical. So we would not be able to tell the difference between mine and my grandfather, right? If we were suspects, we automatically know it's me because my grandfather's been dead for a long time, right? So we wouldn't be able to differentiate between those 2 individuals, which is why we would look at more than 1 STR. So this is an example using a Venn diagram. So let's say we use the TCAT repeat. We're looking for someone who has a 6 and a 7. So let's say our crime scene DNA had a 6 and a 7 STR for TCAT. Right? All the individuals in that circle are a 6 and a 7. But that's not enough, though. There's too many suspects. Now we're going to add STR number 2, which is a GGCC repeat. And we're looking for an individual who has a 19 and an 8. So if we overlap the data from our STR2 with our STR1, well, that gives us these individuals that match for STR1 and STR2. But still, that's 4. Let's narrow it down. We use STR3, an ACC-TTG repeat, and we're looking for an individual who has a 2 and a 10. So when we overlap all of the data for these 3 STRs, we get one main suspect. Right? This is our main suspect. This is not necessarily our criminal or our perpetrator or our mom or our dad or our child. Right? We do need more information than that. For instance, right, you could have just, I don't know, scratched somebody or something before you died, and you've got their DNA under your fingernails. So DNA is not all you need. DNA is a piece of evidence that tells us we need to look more closely at this individual. So we use PCR, the polymerase chain reaction, to amplify those specific STRs. Right? We're looking at those STRs. Now we have to know the sequence of the STRs, and that's important. So we could really only use this for organisms which we know the sequence of the genome, and we can use PCR to frame that sequence and copy it. Right, remembering that PCR relies on a pair of primers. Again, this is review from last week. Right, those are those short, chemically synthesized single-stranded DNA molecules, and you can literally order those from companies. It's going to be a primer to complementary sequences at each end of our target sequence. It will frame them. And then in our PCR, with each round, we're going to double the amount of DNA that we targeted. Right, once we do that, we can take the DNA samples from after the PCR and load it on a DNA gel, an agarose gel and gel electrophoresis. In this example, right, we've got DNA from the crime scene, DNA from suspect 1, and DNA from suspect 2. We run those on our gel, and then what we often would do is use a stain or something that is specific to bind to DNA, and it will allow us, after the gel has run, to visualize the bands of DNA. Right, so here's our crime scene DNA, the band pattern. Right, and here's suspect 1 and suspect 2. Which one matches? Now, it's really important to make sure it completely matches. So sometimes people will look at a picture like this and be like, well, you know, this band matches this band, and that means we need to bring in suspect 1 because maybe there's a partial match. Well, there's not a partial match, right? There's no such thing as a partial match when it comes to something like this, unless you have a degraded sample of DNA, right? Then you get into something like that. So we don't look at anything that's not a perfect match. Right? When we look at our crime scene DNA and suspect 2, it is a perfect match. Right? We would want to know some more about suspect 2. But the other thing you'd want to do is test the DNA of our victim, if there's a victim. Right? You never know if the victim has left that DNA sample from one reason or another. They did not do that on this gel. Right? Our advantages of this method is we are able to increasingly use smaller and smaller and smaller and smaller samples. So the take home of this is just don't do anything bad. Right? So they have retested samples from 20 years ago that were at that point were too small of a sample to do any sort of analysis on, and now they can do it. It's highly sensitive, right? You get rapid results, but you have to actually be able to do it, right? We talk about the rape kits that are built up in storage in a lot of cities, and that's because you need staff to run this. It is relatively rapid once you can do it, but if you have thousands of tests to run, you still are going to take a long time to get through them all. Right, these are the components of PCR that we talked about last week. So gel electrophoresis, just to revisit that. Again, we would have done this in class. You're going to do your samples, you put them in the gel, and what they've shown here is we have loaded them in these 3 wells. We're going to run them using electricity. And normally we would say we're going to run red right, so the red electrode is here, and the black electrode is there. Right, that also means that because DNA is negatively charged, this is positive, and this is negative, right? If you put the gel in backwards, you're just going to run the DNA off the gel. So we'll run it and it'll look something like this. Right? In this case, because this is all DNA, it is separating based on size, because they're all negatively charged, because that phosphate group is negative, and we can look at the patterns that we have in our DNA gel.